Welcome to PG Pathshala Biophysics. This is paper 3, Cellular and Molecular Biophysics. I am Dr. Ruby Dhar, a DBT Biocare awardee, and I work at All India Institute of Medical Sciences, New Delhi. Today I will be talking about Module 17, Methods to Study Cell Motility. Now, in this chapter, we'll try to understand why cell motility is important, what are the various methods of cell motility, and how we can study motility. Motility is a major characteristics of living organisms and can take the form either of movements of cells or movements within the cells. In biology, motility is the ability to move spontaneously and actively, consuming energy in the process. Most animals are motile, but the term applies to unicellular and simple multicellular organisms as well as to some mechanisms of fluid flow in the multicellular organism. Motility is genetically determined but may be affected by environmental factors. There are several methods to understand the motility of cells under the influence of various conditions, and this chapter gives an overview of various technologies developed so far to understand the process of cell motility. Now, just to give a further more introduction about the process of motility, most cells in the body are stationary, but many of these exhibit dramatic changes in the morphology, the contraction of muscle cells, the elongation of nerve axons, the formation of cell surface protrusions, the constriction of a dividing cell during mitosis. Cell motility is needed for some vital physiological procedures amid improvement, for example, cell movement amid gastrulation, tissue recovery, and embryological advancement. A type of cell motility includes the dynamic transport of membranous organelles inside of the cytoplasm and its required distribution of metabolites, hormones, and different materials inside of the cell. Cell motility is one of the crowning achievements of evolution because active separation of chromosome depends on motility of the chromosome, the streaming of cytosol, the transport of membrane vesicles. These internal movements are essential elements in the growth and differentiation of cells, carefully controlled by the cell to take place at specific time and in a particular location. Now, advances in technology development have enabled us to monitor the motility of single cell or group of cells both in 2D and 3D method and also live cell tracking. This following chapter has included the various objectives like the motility, various type of motility, difference between motility and Brownian motion, methods to study bacterial motility, studying sperm motility, various methods of 2D single cell locomotion assays, 2D methods to study group cell motility, then 3D cell motility assays, microscopic methods for studying cell motility, and microfluidics to understand cell motility. Motility as a vital part of living organism. Our license with cell migration as humans begins shortly after conception, accompanies us throughout life, and often contributes to our death. In my introduction, I had discussed various examples which require cell motility in everyday functioning of human body. The importance of cell migration, however, goes far beyond humans and extends to plants and even to single cell organisms. All cells can be considered motile for having the ability to divide into two daughter cells. Now, at the cellular level, different modes of motility exist. There is number one, the flagellar motility. It's a swimming-like motion observed, for example, in spermatozoa propelled by the regular beat of flagellum or E. coli, which swims by rotating a helical prokaryotic flagellum. Now, the second is amoeboid movement, a crawling-like movement which also makes swimming possible. The third important type of motility is the gliding motility, and the fourth type is the swarming motility. Now to further go in detail of the flagellar movement, a flagellum is a whip-like appendage that protrudes from the cell body of certain prokaryotic and eukaryotic cells. The primary role of the flagellum is locomotion, but can also be used as a sensory organ. The prokaryotic and eukaryotic flagella differ greatly in protein composition, structure, and mechanism of propulsion. However, both can be used for swimming. An example of a flagellated bacterium is the ulcer-causing Helicobacter pylori, which uses multiple flagella to propel itself through the mucous lining to reach the stomach epithelium. An example of a eukaryotic flagellate cell is the mammalian sperm cell, which uses its flagellum to propel itself through the female reproductive tract. Now, second, amoeboid movement. Amoeboid movement is the most common mode of locomotion in eukaryotic cells. It is a crawling-like type of movement accomplished by protrusion of cytoplasmic of the cell involving the formation of pseudopodia and posterior uropods. The cytoplasm slides and forms a pseudopodium in front to move the cell forward. And examples are amoeboids, the slime molds, and some protozoa such as Naegleria gruberi, as well as some cells in humans such as leukocytes. Now, sarcomas or the cancer cell arising from connective tissue are particularly adapt to amoeboid movement so that they can lead to metastasis and invasion. Now, next coming up is the gliding motility. Gliding motility is a type of flagella independent translocation that allows the microorganism to glide smoothly along the surface without the aid of any propulsive organ. Gliding motility and stretching motility both allow microorganisms to travel along the surface of low aqueous flames, but while gliding motility is smooth, stretching motility is jerky and uses the pili as its mechanism to transport. Examples are Epicomplexa, a eukaryotic parasite traveling at fast rates, and Myxococcus xanthus gliding at the rate of 5 micrometer in a minute. Now coming next is swarming motility. Swarming motility is a rapid and coordinated translocation of a bacterial population across solid or semi-solid surface and is an example of bacterial multicellularity and swarm behavior. Now, what is the difference between the term motility and Brownian motion? Motility is different from Brownian movement and both have the following characteristics. Now, talking of motility, it's a unique and it's a directional movement of individual strengths from one point to the another. On the other hand, Brownian motion is the aimless and unidirectional movement, and it includes oscillation and quivering motion. Now, next is the methods to study the bacterial motility, or which are the flagellar dependent motility. Now, there are three main methods under this category, and we'll start up with the hanging drop method. Number 2 is the observation of motility band, and 3 is the Bailey's method. So, there are three methods for studying the flagellar dependent movement. Number 1, we'll go in detail about the hanging drop method. This method is used to stain live unstained bacterial cells suspended in water or a broth. Now, in this method, we take a clean glass slide and apply parafilm ring or adhesive tape ring to make a circular concavity. This step is not needed if a glass slide with depression is available. Then hold a clean cover slip by its edges and carefully dab Vaseline on its corners using a toothpick. Now, place a loophole of the broth culture to be tested in the center of the prepared cover slip. Next, we turn the prepared glass slide or the concavity side upside down so that the Vaseline seals the cover slip to the slide around the concavity. Now turn the objective to its lowest position using the coarse adjustment and close the diaphragm and look under the microscope to determine the motility of the bacteria in culture. Now the advantage of the hanging drop method is that it preserves the bacterial cell shape and arrangement. It also allows for longer observation since the drying out process would be very slow. Disadvantage included in this method is it's too risky if we are using a highly pathogenic organism. Now the second method is the band detection method. This is a tube motility medium and a wire loop is used to stab the medium with the testing bacteria. Now non-motile organisms which lack flagella will form a single line of growth, while motile bacterium will grow and make a hazy zone around the stab line. The third method to study the flagellar movement or the bacterial movement is the Bailey's method. Flagella staining by Bailey's method, capsule staining using Congo red and manual staining solution. Now methods to study sperm motility. Analysis of sperm motility begin within 1 hour of specimen collection. A drop of undiluted semen is put into the hemocytometer and a cover slip is put on it. Now we observe under a bright field microscope with very reduced light, and this can also be observed under high resolution video photography or computer assisted semen analysis. Next, coming up to the 2D single cell locomotion assays. Now single cell migration assays are well adapted to study the single cells in inflammatory and immune reactions and tumor spreading. The first method is colloidal gold migration assay. Cells are cultured on a glass substrate like cover slip covered with gold particles. After the required experimental treatment and incubation, cells are fixed, the cover slip is mounted onto glass microscope slides. Computer aided analysis can be used to monitor the images of the tracks made by the cells and measure the area of the tracks. Now the second is live single cell tracking. This consists of automatically recording sequences of frames of living cells in standard cultures by means of microscope equipped with video acquisition system. And now the various XYZ locations of each cells are measured to confirm the cell trajectories. The next method is the Dunn chamber assay. This device is useful for studying chemotaxis properties of a cell. The two device is formed by two concentric wells separated by an annular platform or 20 microns below top of the chamber. The inner well is filled with a control medium and the outer with a medium containing a potential chemoattractant. A radially directed linear diffusion gradient becomes established in the gap. Cells are grown on a cover slip that is inverted onto the Dunn chamber, and those that are over the annular platform can be observed under a phase contrast microscope and recorded automatically by an acquisition system. Now 2D methods to study group cell motility. So far we were studying single cell, now group cell. Chemotaxis under Agarose method. The classical method of choice to study chemotaxis is based on the Boyden chamber assay, but it has its own limitations and are affected by factors such as cell adhesion to the filter material and the tortuosity and size of the pore channel. This method is commonly used to study the migration of leukocytes and utilizes the following steps. Now agarose is dissolved in sterile distilled water and boiled to make a clear suspension. After cooling it around 40 to 45 degrees, the agarose is mixed with equal volume of pre-warmed minimal essential medium containing air of serum. 5 ml of the prepared agarose medium is delivered to each 60 by 15 mm tissue culture dish and allowed to harden. The dishes has to be kept at 4 degrees for 30 to 60 minutes so that it is solid enough for cutting wells. Now 6 series of 3 wells of 2.4 mm in diameter and spaced 2.4 mm apart are cut in each plate using plexiglass template and stainless sponge. Cells should be suspended in MEM and 10 microliter cell suspension added in the center well. The outer well receives the chemoattractive factor in 10 microliter volume. The inner well receives 10 microliter volume of appropriate non-chemotactic control medium like MEM or some containing the vehicle. Next, the dish needed to be incubated at 37 under proper conditions and for proper time. Then after the required incubation time, cells are fixed with methanol, stained with right stain and air dried. The migration pattern are projected onto a white background at 40x magnification. Quantification of migration is done by measurement in centimeters of the linear distance of the cells had moved from the margin of the well toward the chemotactic factor. Now this is the distance A, and the linear distance the cells have moved from the margin of the well to the control media, which is distance B. Now the second method in this group is the microcarrier bead assay. This is one of the first assays used to evaluate cell migration. It's relatively less used now as it has been modified in a more 3D environment. And this assay includes various steps like cells are grown to confluency on a microcarrier bead. Cells coated beads are placed in 24 well plates where they are grown and can be subjected to various treatment. Now after the required incubation time, the beads are removed by suction and washed, and the cells that have migrated from beads to the plastic surface of the well are fixed and stained with crystal violet. The quantity of the migration cells is then evaluated under a microscope. The third method is the ring assay. This assay is also called the fence assay, and the following steps are undertaken to execute this assay. Cells are initially grown inside a Teflon or glass ring in a 6-well culture plate. Once the cells become confluent inside the ring is removed and the cells are washed thoroughly to remove any unattached cells. Cells are treated and incubated for the required time and are allowed to migrate if the cells want to move further. Cells are then rinsed, fixed and stained with toluidine blue and observed under a microscope to visualize the net increase in the total area covered by the cell. The next method is Aggregate Migration Assay AMA. This assay is based on the similar principle as ring assay. The only difference is cell aggregates are prepared by centrifugation and sedimentation and plated onto coated glass plates for migration. Cells are treated and incubated according to the experimental requirements and then stained and assayed as ring assay. Now the fifth assay in this category is the wound healing assay. This assay is based on making a wound after the cells are grown in a confluent monolayer layer in proper experimental conditions. The wounds can be made using a pipette tip, a syringe or a sharp wire. Cells are incubated under the required treatment and can be observed for a prolonged time until the wound mark is healed. The monolayer recovers and heals the wound in a process that can be observed over a period of time under a phase contrast microscope or an automated microscope. Now next is 3D cell motility assay. Now in this category, Boyden method or the matricial invasion is a commonly used method routinely to understand cell invasion. It is performed in 2 wells into which membrane inserts are placed. These inserts are coated with a ECM like extracellular matrix proteins and have pores small enough like 5 to 12 micron size to restrict active passage of cells that have been added to the upper layer. Cells are plated onto the insert well, which in turn is dipped inside another chamber and is filled with the media containing various chemoattractants and are incubated for the required time period. To evaluate migration or invasion rate, filter is removed after the set period, then cells are stained. Cells which have migrated through the filter are then stained with crystal violet and counted under a microscope. Migration is defined as percentage of cells passing through this filter. Now, the next method in this category is collagen invasion assay. This assay consists of initially seeding cells onto a surface of a collagen gel and monitoring the depth of migration of these cells into gel by means of phase contrast microscopy equipped with a computer assisted stepping monitor so that you can understand how much the cells have invaded. The other method is the invasion in organ culture. This assay consists of three-dimensional confrontations between tumor cells and normal tissue, usually done for embryonic animal organ fragments or organotypical human tissue. Pre-cultured organ fragments are converted with aggregates of the tumor cells, first on semi-solid agar to allow attachment and subsequently in a suspension culture in liquid medium for several days. Now, for histological analysis, cultures are fixed and proceeded using hematoxylin eosin and immunohistological staining for known invasive markers. Now, coming up next is the use of microscopy in studying cell motility. Various types of microscopy is being widely used to study cell, and there is a constant evolution in the technology to develop more sensitive and high resolution microscopy. Microscopy is routinely used to understand the protrusion of lamellipodia, filopodia, cytoskeletal dynamics, and membrane morphology of moving cells with high resolution and sensitivity such that the underlying mechanism of protrusion and retraction can be understood in the context of interaction of various growth actins, microtubules, and intermediate filament cytoskeletons. Underlying all direct imaging studies of living cells or organism is the desire to preserve the living object for as long as possible through minimizing of both phototoxic cell or tissue damage and photobleaching of the incorporated fluorophores. So, in this method, the most commonly used is phase contrast microscope, which was discovered by Dr. Zernike in 1935. It uses a spatial condenser and objective lens. The condenser splits a light beam and throws light rays slightly out of phase, and the beam of light then passes through the specimen, and the small difference in refractive index within the specimen show up as different degrees of brightness and contrast. It's a very useful method and is routinely used for studying microbial and eukaryotic motility. Next is the automated microscope system cell analyzer. So, this uses a motorized microscope components and accessories enable the investigator to automate live cell image acquisition and are particularly useful for time-lapse experiments that range in time scale intervals from milliseconds to 10 or hundreds of minutes. Visible responses of whole cells can be yield insight into functional changes that underlie physiological process in health and disease. So, it's a very useful method for live tracking or live seeing the live cell imaging. Next is the centrifuge microscopy, and is composed of a centrifuge and a microscope optical system designed to observe minute objects, specifically living cells, during the application of centrifugal acceleration. These studies include observation of the behavior of cells and cell components in a centrifugal field. It helps in determination of the mechanical properties of the cell surface and cytoplasm. Next, very important microscopy method is the confocal microscopy, and it offers several advantages over conventional optical microscopy, including controllable depth of field. the elimination of image degrading out of focus information, and the ability to collect serial optical sections from thick specimen. The key to the confocal approach is the use of spatial filtering to eliminate out of focus light or flare in specimens, and this can be used for immunohistochemical staining of various proteins regulating motility. Next microscopic method is the SAM, scanning acoustic microscope. A scanning acoustic microscope is a device which uses focused sound to investigate, measure or image an object, a process called scanning acoustic tomography. With this method, intracellular motility of normal and transformed cells of epithelial and fibroblastic origin was investigated. Next is the confocal scanning acoustic microscope, and this uses a wide range of transducer frequencies, providing the flexibility to look at various materials and sites. Low frequency transducers such as 10, 15, 20, 30 MHz allow for higher penetration through materials but low spatial resolution. Higher frequency transducers from 100 to 230 MHz give higher resolution and are used once for microscopy. Through scanning, microscopy detects voids at any depth in the device. Next is the Ibm Intravital Microscopy, allows for direct in vivo visualization of dynamic biological process in their physiological context at high spatial and temporal resolution. This has led to important new insights into immune cell behavior at steady state, as well as their change in behavior during an immune response. Next is the cell traction force microscopy, CTFM. Migration is a vital characteristic of various cell types and enables various cellular functions during development and wound healing. Cell movement can be measured by monitoring cell traction forces, which are generated by individual cells and transmitted to the substrate below the migrant cells. This method has the advantage of directly measuring the cause, that is the cell traction forces of the cell movement, rather than the effect. Added new upcoming technologies, microfluidics for studying cell motility. Microfluidic devices consist of small channels with micrometer dimensions and are being developed for cell migration studies. These devices can precisely configure and flexibly manipulate the various concentration gradients, electric fields, and thus can be used to study the complex mechanism controlling cell migration and motility. It's a very rapid method. Effectors can be studied to study the effect on cell motility. To summarize this topic, cell motility is a very important process in any living organism development and is involved in embryonic development, contraction of muscle, elongation of muscle, tissue repair, cancer cell invasion and bacterial movement. At cellular level, there are four major types of motility, as we discussed: the flagellar movement, amoeboid movement, gliding and the swarming movement. Now there are three main methods to study bacterial motility, which are the hanging drop method and observation of the motility band and the staining by the Bayes method. Now sperm motility is also a eukaryotic example of a flagellar movement, and it can be studied under a microscope, a regular microscope or a high resolution automated microscope. Single cell motility in 2D environment is studied using live cell imaging, colloidal gold and dome chamber method. Now for studying group cell motility in 2D environment, we can use live imaging, wound healing assay, agarose assay, chemotaxis assay, the microcarrier bead assay and the aggregate migration assay. For 3D motility assay, use of Boyden chamber or the matricial invasion assay is very routinely used. The other methods to study cell motility in 3D environment are the collagen invasion assay and the invasion in an organ culture. Microscopy plays a very important role in understanding cell motility, and in this chapter we have discussed various microscopic methods to understand the cell motility and the various cytoskeletal structures involved in controlling cell motility. And the last but not the least, as I said, the technology is advancing, and we are using microfluidics to understand cell motility. And in this, various factors can be studied at a single time point, and it is a very rapid and sophisticated method, and cells can be analyzed both at the single cell level or in a group of cells. And thank you so much for going through this chapter, and with this I end this chapter. Thank you.